Optic genome mapping is a technology that detects genomic structural rearrangement. In a previous video, I talked about why it's important to detect these structural rearrangements, and I briefly mentioned optic genome mapping, OGM. Today, we're going to talk about how OGM works. To detect genomic structural rearrangements, the most classic method is chromosome analysis, and it's used in virtually all clinical laboratories. It requires cells in metaphase. During metaphase, under the microscope, we can recognize one chromosome from the other. For example, we can tell there is chromosome A and chromosome B. And we see that each of them has two alleles. We know one is from mother, that's the maternal allele, and one from the father, that's the paternal allele. However, even the most actively dividing cells spend most of its time in interphase, and that makes it challenging for the detection of structural rearrangement. Because in interphase, the chromosomes are stretched out into long strands of chromatins. So when there is a structural rearrangement, although during metaphase, you can see that chromosome A swapped a piece with chromosome B, but if the cells are in interphase, they look like this, or more like this, because chromosomes have the same color. And this is when optic genome mapping comes in. Optic means that this technology is essentially an imaging assay. It takes high-resolution images of DNA molecule. Mapping means that it takes these images and try to determine where they belong, so that we know if each piece of our genome is situated at the right location or if there is a rearrangement. So it's really like a jigsaw puzzle for our genome. And here is how it works. First, we isolate ultra-high molecular weight DNA. And that's just a fancy way to say that we're going to get the DNA molecule in its natural length as much as possible. Why? Just like playing a jigsaw puzzle, the bigger the puzzles, the easier it is to map them. This is the most critical and the most challenging part of OGM. All the traditional ways of DNA isolation damages and shear the DNA to some extent. None of them are adequate for OGM. So OGM has its own unique way of DNA isolation. So now the ultra-high molecular weight DNA is isolated into a test tube. It is then dyed with a green dye. This green dye recognizes a specific sequence that's distributed all throughout our genome, and each genomic location has its own unique pattern. And the DNA backbone is given a blue color. Now it comes the fun part. This long strand of colorful DNA is then pushed through a series of channels that are positioned like combs, and the teeth of the combs get finer and finer. Finally, in the last channel, the DNA is straightened out and flows through the channel where a high-resolution camera is sitting on top, where it's going to take an image of this DNA and many other DNAs. These images are then processed by a computer software. Much like a human playing a jigsaw puzzle, the software now looks at the unique pattern of the green labeling and try to discern which location this DNA piece is coming from. So now we can tell which chromosome is which, and if one DNA piece matches to two different locations, say two different chromosomes, we can call out a rearrangement. So to recap, chromosome analysis looks at chromosomes during metaphase under the microscope. And by using different banding patterns of the chromosome, it can tell if the chromosomes are normal or if there is a rearrangement. And OGM looks at chromatin using a high-resolution camera during interphase. And by looking at the unique pattern of the DNA labeling, it can discern normal versus abnormal DNA, and it can call out rearrangement. And that's it for today. If you're interested in genetic testing related topics, I recommend my previous video about duplex sequencing. If you have any questions, leave it in the comment below. Thank you so much for visiting, and I will see you next time. Bye.